Explain the components of the nucleotide. So there's a phosphate group, which is attached to the 5' carbon of a pentose sugar, which is attached to a base. There are two types of sugars, the RNA and DNA. The RNA has an extra oxygen on the 2'. That's the only difference. The bases are cytosine, guanine, adenine, thymine, and uracil.
I'm going to explain the structure of DNA. So in DNA, there's a 5' end and a 3' end. The 5' end just symbolizes where the 5 carbon is, which is next to the phosphate group, and the 3' end just represents where the 3 carbon is, which is next to the hydroxyl group. DNA runs anti-parallel, so if it's going from 5' end to a 3' end, the opposite strand goes from 5' end to 3' end this way. In DNA, the nitrogenous bases pair as G and C and T and A. However, in RNA, A and U go with each other.
I'm going to explain the initial components of DNA replication. So what happens is helicase begins to break up the strands, and what it does is the replication fork, like where it begins. This is part of a replication bubble. There are two sides to it, but in this case we're only looking at one side of the fork. So helicase moves through, breaking up the bonds between these groups, thus separating the strands. Now these strands, when come back together, therefore the single-stranded binding proteins will attach to the strands to keep them from coming back together.
First, I'm going to explain the leading strand, which is continuously synthesized throughout DNA replication with no interruption. So first, helicase opens up the DNA molecule, and as helicase opens it up, primase is needed to add the RNA primers, which is needed for DNA polymerase 3 to add the subsequent nucleotides after. It essentially lays a template for the next nucleotide. So here are primase adding this primer and this primer. Now we have DNA polymerase 3 add in the next DNA nucleotide. So here's that. Finally, we have DNA polymerase 1 to replace the RNA primers set down by primase with the matching DNA nucleotides. So here.
So now I'm going to explain the lagging strand, which is different from the leading strand because it doesn't run continuously and they're fragments, which I'll show you later. But we'll start off when the helicase breaks the strands apart like this. The RNA primase and the DNA polymerase only work from the 5' to 3' direction. So it starts here, building the RNA sugars and sequences in this direction, and then the DNA polymerase 3 uses this as a template to build down the rest of the way. Then the helicase continues to move up, breaking these further and further apart like this. And the same process happens. The RNA primase will start here, and it will build the RNA sequences like this, like that. And then the DNA polymerase will continue using this as the template. And then, now as you can see, there's this fragment here where there are the DNA sugars and the RNA sugars, and these are called Okazaki fragments. So what happens initially is the DNA polymerase 1 will go through and it will replace all of these with the DNA sugar. And then, where these Okazaki fragments were, where the RNA sugars and DNA sugars met, ligase will go through and make the bonds between all of them, between these, and then thus binding the entire DNA. Now on both strands there are phosphodiester bonds that are located between these, and they're on both strands, leading and lagging, and that's what holds the DNA together. And the ligase, like I said, will make the bonds through here.